This webinar today is both it is an ELIXIR webinar, obviously, but it's also in association with the ELIXIR Systems Biology Focus Group. And this focus group's objective is to make recommendations to ELIXIR on its future participation and support for systems biology. And this topic that Henning's going to talk about is clearly very relevant to that general area. So the seminar will be given by Henning Hermjakob, as you've heard, and I'm sure many of you know him. Henning leads the molecular system services at EMBL-EBI, which provides a number of services relevant to systems biology, including BioModels, Intact, and Reactome. And his title today is Reproducibility in Systems Biology Modeling Sometimes. I'll pass you over to Henning.
Okay, so good afternoon, ladies and gentlemen, and welcome to the seminar on a study we did on reproducibility in systems biology. And I will have three parts to the presentation. A brief introduction to the BioModels database, which is the parent project to the study, the actual reproducibility study, and then a couple of slides on a larger context in which I hope the results of the study will have an influence on collaboration between different model repositories, namely ModeleXchange. Okay, so one slide introduction to what's mathematical modeling. The aim of mathematical modeling in systems biology is to develop a formal representation of a biological system, which ultimately will allow to do in silico predictions, which then, of course, hopefully are verifiable in wet lab experiments. And this is to cut down on wet lab experiments and make them more targeted, and of course to help our understanding of the biological system. So starting from the biological system and experiments which generate data on the system, mathematical representation is developed, and we are in this study only considering ordinary differential equation-based models. There's many different modeling approaches, but we will focus here on these ODE models. So after developing this formal representation of the biological system, then you of course want to do predictions, and you then try to validate them in the wet lab. And this is typically a cycle where iteratively the models are improved and our understanding of the biological system is improved with it. BioModels comes in as both a resource, which is a starting point for modeling, in the same way that you start from what is already established in the literature when you work on any scientific topic. We provide BioModels as a resource for taking models which are relevant to your research topic as a basis for either inspiration or direct further development. And of course, to do that, we need to capture models which are published by the scientific community in the BioModels database. And the aim is to support modelers in the modeling process and to implement the classical standing on the shoulders of giants, where new models are built on the experience of previously developed models, so that ultimately the progress in the domain is faster. This is the homepage of BioModels. We have the usual Twitter feed, the various entry points, browsing by different parameters, and we typically provide a model of the month to have to highlight interesting models which have been curated in the last month or so. We focus on on, as I mentioned, modeling approaches, or the modeling approach of ordinary differential equations. There's other approaches, logical modeling, PetriNet, constraint-based, many others. And we focus on formally representing these models in the Systems Biology Markup Language, SBML community standard, and related standards. But again, there's others which we can also represent in BioModels, but we focus on SBML and related. So a typical curation procedure for a model to get the model into the database is roughly outlined on this slide. So starting from a biochemical network, for example, it can also be something like, let's say, virus spread in population. The approach is agnostic to scale. This network is then represented in a set of mathematical equations and associated data, in particular constants and concentrations for the model. And these might either be the formula might either be directly taken from the paper by the curator or submitted to us as an already prepared direct model submission. And in either case, and then we will only have to validate these. Then we ensure the representation in SBML, either because it's already available in this, because we directly encode in SBML, or through conversion and then manual adaptation. Then this model is so-called run. So a model simulation tool like Copasi is used to execute the model. And that then provides simulation results, for example, time course. We choose our models both based on curator expertise, on collaborations with partners in the community, based on funding, and on direct submissions. So there is no specific focus of BioModels. BioModels is systems agnostic, and the choice of models depends on these factors which I've just mentioned. It can also be hot topics of the year, like our current curation effort for COVID-19 related models, which is done together with a group of Dagmar Waltemart, where we are trying to provide a set of high quality curated models in SBML, and we provide these on a specific web page, subpage of BioModels. And this is an ongoing effort. Any contributions are highly welcome. So once the models are in BioModels, of course, we want to make them findable. This is a typical search results page of BioModels. You get a list of the models, the typical faceted search results where you can narrow down your search results. And then you can click on the individual model and you get views like this. The overview provides a short description, the associated publication of the model, and some administrative parameters, key parameters of the annotation. We provide the actual model files in a variety of formats, different SBML versions, and additional files, which, for example, describe what we've done in the execution of the model. We provide a detailed history of the data set, the model in our resource. So what are the changes over time? And overall, BioModels is fair. Models are findable, of course, through BioModels search itself, through the EBI search, through omicsdi.org, which is a cross-domain search tool which allows dataset-specific search across transcriptomics, proteomics, and many other omics approaches. And we've also included BioModels models in there. And through omicsdi, we are also making the models findable as in the Google dataset search through export based on schema.org. And there's also work going on specific export in bioschema, which is one of the key ELIXIR activities. Everything that BioModels provides is as open as possible. All the models are released under CC0, and the code is entirely open source. And we try to support interoperability as much as we can. As mentioned, we focus on curation of, or when we do curation, we do it exclusively in SBML. We also provide other models as a repository, but our curation efforts are focused on the SBML community standard. And we are actively curating into this. And of course, the aim is to provide reusable models. All the models in the curated section of BioModels are tested. As I mentioned before, they are run through a simulator and they are validated against the associated publication. I'll come back to this a bit later. And BioModels is an ELIXIR deposition database. So thank you for the support to the ELIXIR community. BioModels is widely used in the domain, both for model discovery. In 2017, there was a large-scale survey in the domain on what is the most frequent data source for models people use in their research, and BioModels was very prominent in this. And it is also very popular for dissemination. So here the question was, which resources do you use to distribute your model? And so BioModels was the most used repository, but keep in mind here, supplementary material, GitHub, and personal website are all more used than BioModels. I'll briefly touch on that later on. So there's definitely still room for improvement. So far, I've focused in this description on the curated section of BioModels. This is where the validated models are, which is here this blue area, which contains now just under 1,000 models. And we have another section, the non-curated section. And these are models which are also syntax validated, but they are not semantically enriched. So they are not annotated systematically, for example, with UniProt terms, KB terms, and so on. And they are not validated in the way I mentioned and which I will describe again in the future, either because the validation process was not successful or because we simply haven't had the time yet. And this section also contains models which might be perfectly fine, but which are not based on ODE, which are not ODE models, and because we provide less support for other representations, they also will be kept in the non-curated section. And you see a brief view of how models distribute over species, and as expected, human is the most frequent topic, followed by the typical list of model organisms. Okay, so this was a brief overview of BioModels and what we do in the BioModels database. Now to the specific study. We wanted to assess the reproducibility in systems biology models. And I first have to use one slide on definition, because there's quite different usage of terms in different domains. So in systems biology, typically replicability or repeatability is the ability to use the same code provided with a manuscript in the same software to reproduce exactly the simulation results. So essentially this is, if you run a Docker image which has all that was relevant for that model and it actually works, then you can say you have successfully replicated or repeated this experiment. But of course, this might be not very satisfying. It might be still just, let's say, a binary code which you can't modify or access. So really what we wanted is to assess reproducibility of a model as it is presented in a paper. So the ability to recreate a model without the original code, or at least with using a different approach in the actual simulation run. So this is what we did. We encoded the model equations in SBML, got the parameters from the paper or supplementary material or from direct author submissions. Then we ran the simulations, typically in CoPaRC, one very widely used community tool. And as a key point, if the original simulation was done in CoPaRC, then we systematically use a different simulation tool to make sure that it's actually independent of the tool used in the publication. And we considered a model reproducible if at least one simulation figure from the publication could be reproduced. And we would accept minor deviations if they did not affect the scientific conclusion of the study. So fit, which is not totally perfect between our simulation results and the published figures would be okay. But let's say if the amplitude or the periodicity of a time course is twice, then clearly this is not acceptable. The emphasis is also on one figure. So as a working hypothesis, we assume that if with a given model we can reproduce one figure, then we can probably with more effort also reproduce the other relevant figures, but at some point we had to limit our effort. We can't redo the paper for every publication. So if that worked, great. The model was classified as reproducible. If not, then we try to fix problems. And this is in a first step based on purely curator expertise. And very often there are minor things like there's a minus or a missing minus in front of an equation or a term of an equation. Or there's an error in the units like microliter versus milliliter. And these are often relatively easy detectable and can be fixed by the curator. And if that was possible, then we would consider the model still reproducible. If that didn't work, then we would try to contact the authors and then try to get the model to a reproducible state with help from the authors, typically by asking for clarification on the structure of the model, so the actual equations, or very frequently by getting the parameters which were used to run the simulation. And again, then we would, if that worked, then we would consider the model reproducible. So this is one example for the comparison between the figure in a paper and our simulation results. So this is figure 5 from this publication. And this is what it looks like in BioModels in the entry for the corresponding publication. Under the curation tab, you will always find the results of the simulation efforts. And there's a comment which then describes what we have done. So here we have reproduced the plots of figure 5. The curves correspond to the magenta colored curve in the original publication. And if you look here, then you can see that indeed these curves are identical, and we consider this a successful reproduction. We also show how we have done this with which tool, which version, etc. And this is available under the curation tab for every model in the curated section of BioModels. So any curated biomodel has been executed and works. For the study, we curated altogether 455 kinetic models of biological processes from peer-reviewed articles in 152 journals, which were published over the last 40 years. So from 1980 to 2020, so a very broad range, but obviously with a strong bias to newer models. They were curated as part of the normal BioModels activities from beginning of 2018 onwards until April this year. And I should emphasize this was not a randomized sample as I described earlier. This was based on normal BioModels curation priorities, which are based on what comes in as direct submissions, on funding from specific projects, collaborations, etc. But given that BioModels is the largest repository of ODE-based models, at least in the public domain, and these 455 models, that's about 1/4 of the entire content of BioModels. This is a significant portion of all publicly available models. So we believe we have done a really good cross-section of available models. And these are the results. Quite exactly half of all models were directly reproducible. So without any further intervention, we could recreate at least one figure from the paper, and we assumed that the model really matches what was used in the paper. So you can say the glass is half full or half empty. You can say half of all models failed reproduction, and I think that's really the case. You would hope that it's 100%, which are reproducible. But as I mentioned, we then continued to try, and of the among the 49% of models which failed reproducibility in a first round, we could then still, with empirical correction, so basically curator expertise and try and error, rescue another 9%. And with author support, we could rescue an additional 3% of the original models, so that we could overall classify as reproducible somewhat around a bit less than 2/3 of the models, or a bit more than half of the models, somewhere in between. And this is currently accessible as a bioRxiv paper. And the tweet on this was the most successful BioModels tweet ever, with 50 retweets and 100 likes. So we gather there's quite some interest in the study in the community. So what did we do in the case where we could not reproduce the model? As I mentioned, we first tried through curator expertise, and then we tried to contact the authors. And this is where it really gets a bit sad. Only 30% of all authors try to contact responded at all to our requests for further clarification to help make their model reproducible. So that's, while not uncommon in the domain and in other curated resources, but still quite a sad result. And in about half of the cases where the authors responded, we could actually then reproduce the model with the author's help. So it's really worth it. You could also ask why it didn't work in the other half. And so this is often then that we first ask for a parameter, it still didn't work, then we send all we have, and then the author didn't respond or something like this. So at some point then the email thread petered out, and we had to at some point close the books and say, okay, we did not manage to reproduce this model. There was a connection to age of the publication, but a bit to my surprise, the reproducibility does not dramatically decrease with the age of the publication. as seen in this graph here, although of course author interaction is practically impossible once a paper is 5 years old, which is not really surprising. When the PhD or postdoc has gone, then it's very unlikely that you can get any feedback from the authors. There's no clear pattern by journal. We didn't systematically try to relate this to impact factor or so, but we didn't see any clear patterns where specific types of journals were publishing more reproducible or less reproducible results. Although, of course, there are some cases where journals have, at least so far in our hands, only provided non-reproducible models, which is a bit dubious. But again, the sample sizes here are relatively small. But of course, we would like this much more to be on the green side. We systematically tried to note down the reasons for non-reproducibility, which of course is not always easy. So we have this classification. Key reasons were missing parameters, missing initial conditions, structural inconsistencies in the model. And then there's of course also a large part of attempts where the reasons for non-reproducibility is not clear. If you just can't reproduce the figure, although you've done your best, sometimes you can't classify what really the problem is. So what happens with these models? Ultimately, this is about reproducibility. Is our study reproducible? Yes, at least it will be when it is published, I hope. So all the reproducible models are now in the manually curated section of BioModels. And I've shown before how they are represented. You can see the curation tab for each of the manually curated models. So they are already fully available in the public domain. The non-reproducible models are in the non-curated part of BioModels, but they are not flagged. They are mixed in with models which are simply not yet curated due to the lack of resources, with models which are non-ODE models, etc. And so there are two reasons for this. One is we would first like to get the study published before we make it completely accessible to everybody, because there is quite a high interest, let's say, in this area at the moment. But also from the beginning, when we set up BioModels policies a long time ago, we decided not to highlight the models where the curation failed, basically to avoid a naming and shaming. And this is something where we would be very interested in community input, if we should come up with some kind of stars system or whatever to highlight successful models, which we are doing, but also to, let's say, warn of models where we could not reproduce them. Because there is it's not mandatory to submit a model when you publish it. It's not mandatory to submit it to a public resource. We have so far refrained from this naming and shaming simply because we think that an accessible model is still better than a not available at all model. But this might be reviewed also in the context of the progress of fair publishing of results in science in general. So that's definitely something which might be an interesting discussion topic afterwards. And I would be interested in feedback. So what can we do? We are not the only ones who are working in this domain. And in particular, there is in the US, the large Center for Reproducible Modeling by Herbert Sauro et al. And they have almost at the same time where we made the study accessible, they provided two publications, one which is a lot on best practices, available tools, and so on. And one is on what to do concretely to improve reproducibility. And the approach they are proposing is that one, and they are trialing this now, is one curator, or that you can have a specific reviewer who is doing a review from a reproducibility perspective and is sort of only specializing on that. And that is clearly one good approach. So there's quite a lot of good material in these two publications. As a lesson from, as a lesson from the study, what we propose is a very simple 8-point scorecard, which we hope can be used by authors, editors, and reviewers. Personally, I don't have the best experience with very complex validation and assessment schemes. And the more complex a system is that puts additional quote burden on an author, the less likely it is that it gets properly implemented. So we try to have something very simple in the form of the scorecard, which basically just provides tick boxes. Are the actual mathematical expressions available in the manuscript or supplementary material? Are the initial conditions provided? Are the simulation conditions, like changes in parameters during the simulation run, provided? Are the models in their encoded form publicly available? Are they disposited in a public database? Are they encoded in a community standard like SBML rather than in my favorite programming language? Are the models well annotated so that they are also findable with sub-aspects of the model, like what are the proteins and chemical entities which are under consideration and occur in the model? And finally, are the numerical results, not only the figures, but also the numerical results of the simulation, shared publicly along with the model code? So if just this scorecard would be used systematically, we could already go much further in terms of reproducible models in the public domain. Okay, so this was really the essence of the study. Now I would like to put this briefly in a bit bigger context. Namely, BioModels is not the only model repository in the domain. There's a lot of repositories with different focus, sometimes on modeling approach, sometimes on the systems which are modeled, sometimes on the representation of the models. And all of these work independently, and they might have different approaches to both model retrieval, model dissemination, but also to policies to support reproducibility, although all of them will want to support this in some aspect. But this leaves the user easily a bit perplexed on where should I submit my model? How should I submit my model? Where do I get my model from? And so they really often compete in their approaches. They might collaborate with various journals and implement with these journals different approaches to support reproducibility. There is no common search at the moment across all model repositories. And a big point and a key point for us, having spent a lot of time on the study, is curation of non-reproducible models might be done many times by different repositories and scientists in academia and industry. So if there's a nice, attractive model on a hot topic published in the scientific literature, then BioModels might try to curate it and fail. And then repository X, repository Y also have independent teams. They might find this attractive and try to curate it and fail. And individual scientists might try the same, both in academia and industry. So the same poorly provided and perhaps irreproducible model might be tried to be reproduced many times. And that is a huge waste of resources in the public domain and in industry. And most other domains have addressed these challenges already, long before systems biology. From my own experience in the protein interaction domain, interaction databases have started in 2005 to coordinate their efforts and had the first big publication 2012. Proteome Exchange has unified the repositories for proteomics, again, in activities starting around 2005, 2007, and with a big publication, first big publication in 2014. These are heavily well cited, and both of these consortia are still very active nowadays and have really helped to unify the domain towards more consistency in providing data in their respective domains. And this is really something which I hope we can achieve also for systems biology. And we've started this in currently very loose collaboration ModeleXchange, where we aim to have together across model repositories, well, a web page once we have something to show. We have an advanced draft of a consortium agreement, how we interact with each other. We have advanced discussions on common metadata standards for annotation of the available models. We still need to work on a central model registry. So to really provide, I have tried to curate this model and failed. So basically don't do it or try to do something cleverer than we have done. I think this is a key element which we have not yet developed. We'll need an announcement channel so that in one, from one channel, let's say a Twitter tweet feed, you know, now there's a new model from this collaboration, and then of course where to find it. And of course, we need a central metadata search where with one search entry point, you can access systematically models from all these resources based on their metadata. And then you are guided to the right repository. And we have a current prototype based on OmicsDI, where BioModels is already included fully in OmicsDI. And on a prototype basis, we have records from Physiome, the Physiome project, Cell Collective, and Ferdem Hub, also in the same framework, so that we have quite a spread already geographically. Physiome is based mainly in New Zealand, Cell Collective in the US, and then of course there is some heavyweight in the UK, and Ferdem Hub, which is between the UK and Germany. And of course, we want to publish this whenever we have something to show. And we've worked on that, starting systematically at Combine, the community conference for the modeling community in July 2019 in Heidelberg, Harmony in March 2020 in Hinxton. This was the last meeting which still happened on campus, face-to-face in Hinxton. We went from the meeting basically in lockdown. And the next event where we will continue to work on this is Combine in October, which of course will be virtual. And with that, I would like to acknowledge the BioModels team, which has done all the curation, the developers, which have done the technical basis for this, the Abraham Institute, Krishna Tiwari is based there and has done, was the first author of the study, and all the ModeleXchange partners, of course, most importantly, submitters and users, alumni who have contributed to this, and of course, the funders. Main funding for this came from the IMI initiative, from BBSRC, and from EMBL core funding. Thank you.